U gaat staan. U kunt wel plaatsen.
I open this academic session in which Ms. Slenter will defend her academic thesis, Symphonies of Metabolomics, utilizing graphs as sheet music to orchestrate biochemical interactions relevant in health and disease. I welcome you and wish you lots of luck, of course supported by your paranymph. I also welcome your supervisors, Professor Evelo and Dr. Willegaard, of course the members of the opposition who you will meet later, and of course all the audience on site and online. But before we start the opposition, I'll give you the opportunity to tell your family and friends something about your thesis. And you do that in Dutch.
Yes. Geachte familie, vrienden, collega's, studenten en iedere ander die hier is en interesse heeft in mijn werk. Ik ben heel blij dat jullie er zijn. Het is een lange rit geweest, dat weten jullie ook allemaal. En dit praatje ga ik in het Nederlands doen, want mijn collega's weten als het goed is wat ik gedaan heb. Dus het hoeft niet in het Engels. Waar heb ik nou aan gewerkt die 7+ jaar aan metabolomics data? En dan vragen jullie misschien af, ja, wat is dat überhaupt? Nou, als we dat linken aan een misschien iets bekender woord, metabolisme, oftewel stofwisseling. Dus dat zijn alle reacties die plaatsvinden in ons lichaam. En die reacties die kun je zien als een soort van fabriekje. Er komt iets in de fabriek, er vinden wat processen plaats in de fabriek. En als die processen goed gaan, en er is winst, dan kan die fabriek ook groeien. Kunnen misschien nog meer processen plaatsvinden en uiteindelijk komen er ook producten uit. En ik heb hier voor een fabriek gekozen omdat ik scheikundige technologie als bachelor heb gedaan, uiteraard. En er komen nog meer dingen terug vanuit mijn academische pad in deze presentatie. Dus deze reacties die plaatsvinden, die gebeuren ook in ons lichaam. Maar wat zijn die reacties nou eigenlijk? Stel je voor, je werkt in een autofabriek, we maken auto's. Deze auto heeft nog geen functie, het is alleen maar een casco. Daar moeten wat dingen aan worden toegevoegd voordat de auto echt kan rijden. Wat we nodig hebben bijvoorbeeld is een dak. Nou, toevallig is vandaag lekker weer, maar in Nederland regent het vrij veel, dus een dak is best handig. Nou, dat voegt de eerste monteur toe, die zet het dak erop. Maar die auto rijdt nog steeds niet, er zitten geen wielen in, dus we moeten ook wielen toevoegen. Dat wordt door een andere monteur gedaan. En dan hebben we een auto die echt kan rijden en die zich kan verplaatsen, die een functie heeft. En bij metabolisme kijken we niet naar auto's of naar monteurs, maar we kijken naar moleculen. En deze moleculen worden in elkaar omgezet door verschillende enzymen, die we hier bij de pijltjes zien staan. Als ik dit nu zo laat zien, zal niet iedereen meteen zien welke reacties hier plaatsvinden. Want we hebben bepaalde expertise nodig om deze informatie goed te begrijpen. En als ik dit laat zien, dan zullen er vast heel veel van jullie zijn die zeggen, ja, dat is een muzieknoot. Die herken ik wel. Mensen die iets meer kennis hebben, die iets meer expertise, zullen misschien zeggen, oh, dat zijn 2/8 noten. Er zit ook een lengte aan hoe lang die noten duren. En als ik er dan ook nog eens een bepaalde context bij zet, dan zullen er zelfs mensen zijn die zeggen, dat is een E of een Mi en een Fa of een Fa in de G of vioolsleutel. Er zijn dus verschillende niveaus waarop we deze informatie kunnen duiden. En hetzelfde geldt voor metabolieten. Als ik dit laat zien, zullen de meesten van jullie waarschijnlijk wel zien, dit is een molecuul. Maar wat voor molecuul is het? Nou, de mensen die scheikunde hebben gestudeerd zullen waarschijnlijk zien, oké, dit is een organische verbinding. Er zitten alleen maar koolstoffen, zuurstoffen en waterstoffen in. En als je nou nog een beetje context geeft, dat dit molecuul gamma-hydroxyboterzuur heet, dan zullen de mensen die forensische wetenschap hebben gestudeerd, waar ik mijn master in heb gedaan, zeggen, dat is GHB. En GHB, zoals sommigen van jullie misschien wel weten, is een drug die wel eens in Nederland gebruikt wordt, maar die dus ook een functie heeft in ons menselijk lichaam. En nu gaan we dan kijken naar wat nou als we deze metabolieten in een bepaalde reactie aan elkaar verbinden. Dus ook hier, wat we dan eigenlijk doen, is die informatie, die context koppelen, zodat een computer beter begrijpt wat hier staat. Want deze moleculen, dat snapt de computer niet direct. Maar als ik er een naam aan geef en als ik er dan ook nog een nummertje aan vastkoppel, oftewel een identifier, dan kan de computer deze informatie begrijpen. Wat jullie waarschijnlijk wel zien is dat sommige van deze nummers heel lang zijn en andere heel kort. De ene begint met een P, de ander met een Q. We hebben eentje met een cabby dubbele punt, een EC dubbele punt. Ja, wat is dit nou eigenlijk allemaal? Nou, daar heeft een computer dus ook wel moeite mee. Een computer kan niet deze informatie aan elkaar koppelen zonder dat hij weet hoe hij dit moet koppelen. Wat wij doen in deze reactieschema's die we maken, is dat we alles organiseren en aan elkaar koppelen. Daarna zetten we ook die enzymen erbij, die zorgen dat deze reacties plaatsvinden, de monteurs. En we zetten ook de identifiers, die nummers erbij en de database waar het vandaan komt, of de databron in het Nederlands. Want zonder dat we deze bron hebben, weten we niet waar de informatie vandaan komt. En daarnaast moeten we ook deze informatie samen koppelen. Dus niet dat elke databron op zichzelf staat als een individueel eilandje, maar dat we deze informatie koppelen en synergie creëren op deze manier. En daar heb ik ook wel een flink deel van mijn tijd aan besteed om die metabolietendaad die er is, om die beter te koppelen. Nou zit ik zelf in een harmonie, zoals waarschijnlijk velen van jullie weten. En ik speel daar dwarsfluit en piccolo. En dit is een muziekstuk wat ik echt ontzettend mooi vind. De 4 jaargetijden van Vivaldi. Maar daar zijn 4 delen van. En er zijn heel veel verschillende instrumenten die daar een rol in spelen. Dus dit is een voorbeeld van hoe het eerste seizoen lente eruit ziet. Als we dan gaan kijken naar het tweede seizoen zomer, gewoon iets wat ik online kon vinden, ziet er weer anders uit. En dan is er nog één voor de herfst en weer een andere voor de winter. Dus wat we eigenlijk nodig hebben is een overzicht. En dat is wat een dirigent heeft. De dirigent heeft een grote muziekpartituur voor zich en kan voor elk instrument zien wat ze wanneer moeten spelen. En in welke volgorde die stukken gespeeld moeten worden. Ja, je kan ook beginnen met winter en dan zomer, dan herfst en dan lente, maar dat voelt niet heel logisch. Daarnaast, wat je ook wilt als muzikant, is een partituur of een muziekblad per instrument. En je moet kijken, kan ik wat hier staat spelen in mijn orkest? Ik zit zelf in een harmonie, daar zitten geen violen in, normaliter. Dus alle vioolstukken, ja, daar kunnen wij vrij weinig mee. En hetzelfde geldt misschien voor een symfonieorkest. En wat als laatste heel belangrijk is de snelheid. In welke snelheid moeten deze muziekstukken gespeeld worden? En die zijn meestal anders per stuk. En daar hebben we kennis van nodig en een overzicht om dat muziekwerk op de best mogelijke manier ten gehore te brengen. Als we kijken naar metabole reacties, we willen die informatie met elkaar koppelen. En dan ga ik weer even terugkijken naar dat voorbeeld van GHB. Voor deze 3 metabolieten vind ik dit reactieschema. Maar ik vind ook deze online. En ik vind er weer één. Die lijken vrij weinig op elkaar, behalve dat ze 1 term gemeenschappelijk hebben. Nou, hoe zorgen we nou dat we die informatie bij elkaar leggen en kunnen integreren? Ook daar hebben we weer een overzicht nodig, zodat we weten welke verschillende reacties er allemaal plaatsvinden en in welke volgorde die reacties ook plaatsvinden. Wat ook heel belangrijk is dat we bewijs hebben voor de reacties. Welke reactie gebeurt wanneer en welke gebeurt niet. Wat heel belangrijk is dat we die informatie van die identifiers, die numerieke codes goed kunnen koppelen aan elkaar. En als laatste de reactiesnelheid. Hoe snel vinden deze reacties plaats? Dat is ook heel belangrijk voor de uiteindelijke interpretatie. En waarom wil ik dat nou eigenlijk allemaal doen? Nou, stel, je gaat naar de dokter, je hebt bepaalde klachten en de dokter zegt, oké, ik ga een bloedmonster nemen en dan gaan we het gehalte bepalen van een aantal metabolieten. We kunnen je ijzergehalte bepalen, we kunnen naar een aantal dingen kijken. En daar komt uit dat een aantal wat hoger zijn dan verwacht voor gezonde mensen, een aantal wat lager. Maar als we dat nergens aan kunnen verbinden en we weten niet echt welke ziekte daarbij hoort, dan moeten we dus toch gaan terugkijken naar die metabole reacties. Maar zoals ik net al liet zien, die zijn heel erg verdeeld. En om die informatie goed inzichtelijk te maken, willen we eigenlijk al die verschillende reactiepaden met elkaar verbinden. En dat kan. Daar heb ik een methode voor bedacht en dat staat in hoofdstuk 4 van mijn thesis. En er zijn al een aantal andere mensen die ook bepaalde methodes bedacht hebben. En wat het mooie hiervan is dat wij 3 verschillende informatiebronnen aan elkaar koppelen en echt specifiek gericht op metabolieten. En dan kun je zo'n schema krijgen waarin je dus die 4 veranderde biomarkers, zoals ze wel heten, aan elkaar kan verbinden. En dan valt jullie misschien op dat een van de pijlen een stuk groter is dan de rest. Dat is waar die reactiesnelheid om de hoek komt kijken. Want die reactiesnelheid kun je je voorstellen als een snelweg. Als de snelweg heel smal is, kan er maar 1 auto tegelijk over. Als je een hele brede snelweg hebt, kunnen er 3, 4, 5 auto's tegelijk over. Dus hoe sneller deze fluxen, zoals het heet, zich verplaatsen door zo'n pathway, door zo'n reactieschema, dat kan ons helpen om te begrijpen waarom sommige gevonden worden in een heel hoge concentratie en andere in een heel lage concentratie. En dan wil ik afsluiten graag met het stukje voor de klinische relevantie. Want dit klinkt vooral als iets wat leuk is voor computers, maar waarom doen we dit nou eigenlijk? Nou, wellicht hebben jullie wel gehoord van deze actie, Serious Request van 3FM, wat ook heel mooi koppelt aan het muziekgedeelte van mijn PhD. Zij hebben dit jaar bedacht om voor het goede doel MetaKids geld in te gaan zamelen. En MetaKids is een stichting die onderzoek naar metabole ziektes wil mogelijk maken. Dus ziektes die gerelateerd zijn aan het metabolisme. Ja, waarom is dat nou belangrijk? Nou, als je een metabole ziekte hebt als kind, ga je er heel waarschijnlijk aan dood. Gewoon daarom. En in Nederland zijn er meer dan 10.000 gezinnen die 1 of meerdere kinderen hebben met een metabole ziekte. Elke ziekte individueel is waarschijnlijk heel zeldzaam, maar als je kijkt naar het totaalplaatje, alleen al in Nederland hebben we zeker 10.000 patiënten, families die we zouden kunnen helpen. Daarnaast wordt er elke dag, en dit is een grove schatting, 1 kind geboren met zo'n metabole ziekte. Die zijn op dit moment heel moeilijk te diagnosticeren. Soms is er een proces van maanden tot soms wel jaren. Wat we eigenlijk willen is dat we dat diagnoseproces sneller kunnen maken. Want zonder diagnose is een therapie bedenken heel moeilijk. En het enige om daarmee te helpen is eigenlijk onderzoek doen. En dat is dus wat ik probeer te doen, mijn steentje hieraan bij te dragen. Dan ga ik weer even terug naar het overzichtsschema wat we net hadden. En dan zie je hier dat stukje in het roze wat erbij is bedacht. Dat is dus 1 van die metabole ziektes. En wat ik gedaan heb is dat aan de reactieschema's koppelen die we hebben, zodat we de patiënteninformatie beter kunnen visualiseren. En hopelijk kan dit in de toekomst leiden tot een snellere diagnose. Ook hier is 1 hoofdstuk van in mijn thesis aangewijd. En daarnaast kunnen we dit ook hergebruiken in andere onderzoeken. En ik ben heel blij met de samenwerking met het ziekenhuis hier in Maastricht, die de data van de patiënten beschikbaar heeft gesteld, zodat ik deze analyse kon bouwen. Nou, ter conclusie van dit verhaal, het is heel belangrijk om specifieke kennis te hebben om deze informatie goed te kunnen duiden. Daarnaast is het ook belangrijk dat we die databases en databronnen en de informatie die daarin zit goed kunnen koppelen met elkaar. Wat we ook heel graag willen doen en wat ook gelukt is, wat we laten zien in deze thesis, is dat we die metabole reacties kunnen verbinden en ook kunnen verbinden met de informatie die er is van patiënten. En als laatste kunnen we deze informatie gebruiken om stofwisselingsziekten te onderzoeken. En hiermee wil ik graag mijn lekenpraatje afronden en het woord weer teruggeven aan de prorector.
Dank u wel. Thank you very much, Ms. Slenter, for an interesting presentation. We will now start the opposition, and the first opponent is also the chair of the assessment committee, and that's Professor Shiri Svetlov. And she's professor in hepatic inflammation and metabolic health in the Nutrim department at the University of Maastricht. Professor Shiri Svetlov.
Dear candidate, first I would like to congratulate you and your promotion team for your excellent work. Your work definitely contributes a lot to our, provide us with tools that can definitely help a lot in our scientific work. I'm very much impressed both by your presentation and also by the deep reflection that you had on societal impact in your book, and especially in the impact paragraph. I find it very exceptional. So compliments on that. And I would like to start by asking you in the direction of your application of the tools that you developed. Of course, there's a lot of applications, but you focused a lot on the diagnostic part. And first, I would like to understand whether you see that as a direct application or indirect, and also whether you find it as tools to identify novel biomarkers, or is it actually just to, for example, per patient, to have a better diagnosis for the patient itself?
Highly esteemed opponent, thank you for your kind words and your questions. There are, I think, a plethora of ways to use the information that I've now collected and put together in this thesis. For me, the reason to go into the diagnostic field, that route, is because there could be a direct application, which I really like. But all the information that we collected is shared open source. So I would actually be very happy if other research groups can use this information, maybe to even discover novel biomarkers. For that, I think there are also lots of other techniques there. I think the main example that I'm showing here is also how to visualize the data, and that is sometimes missing from other tools. So you can do a lot of statistical analysis, but the visualization of the metabolic reactions and how everything is linked, I think that is something that is also very important here.
Yeah, so for this part, I understand the visualization when you look into patients and you want to understand the patients, but for identifying novel biomarkers, it's a little bit harder for me to understand, because what I want to understand is what is the difference, for example, if I sit as a researcher and I get, let's say I do metabolomics, proteomics, I do all types of data analysis on a core that I have, rather than if I have it in a visualization. What is then the added value apart from that, of course, is nicer? Is there something that makes it easier to identify?
Yeah, so I believe especially with metabolomics, it's often very difficult to identify the specific molecule that is measured in these techniques. But by combining it with pathways, we could give it a tentative identification, and instead of saying it's mass 361, we could say we see something in the substructure that is very similar to this other compound. And that compound is a known biomarker because it interacts with a certain enzyme. And if we can actually combine that information from the pathways to substructures, which is something I couldn't get to, unfortunately, during my PhD, but it was already quite long, I think, then we can maybe use that information to actually identify which specific metabolite we're talking about, because having a mass could be used as a biomarker, but in another lab, maybe a slightly different mass is measured, and then it gets very difficult to interchange this information between laboratories.
Okay, yeah, so it's mainly about the combination of the data together. And, but I also think it's very nice in the diagnosis, a great tool, and I think you showed it very nicely in chapter 7, how you want to use it for a diagnosis of a specific patient. You showed it in relation to inherited metabolic diseases, to understand better the differences there. I first want to ask you to reflect on the results, because you tested, of course, it's just the proof of concept, I understand it. But were you happy with how successful you were with the diagnosis based on this, or did you expect the success rate to be higher because you have now this tool?
I was hoping indeed that we could diagnose a little bit more compared to the existing methodology, but at least it didn't get worse. And I think the current diagnostic pipeline does something similar, but without doing the visualization in a semi-automated manner. So there's a lot of manual work for the clinical geneticist, which is now a lot easier and takes a lot less time. But there are a few diseases where with the metabolomics data we had, we couldn't get to a diagnosis. And that can either be because we are lacking information in the metabolic reactions, or because these are targeted metabolic assays that they measured, the metabolites that are measured there are not related to the disease, or missing a whole other component, epigenetics, protein-protein interactions, which is something we could combine in this model. But if I don't have the data, then it doesn't mean a lot. So that's also something we're discussing with clinical genetics. For these very specific cases, how could we maybe help them in the future? And then, indeed, the data combination that I did with the pathways was not the way to go, but it's maybe a start, and then to extend it with other data.
But just for me to understand, how will it be in the future? So a clinician is sitting, he's plotting all the data of a specific patients on the computer, and then he sees all the patients. So how do you see the daily application of this? Will it be more for scientists to look into these pathways and try indeed to identify and to understand the diagnosis better, or do you see that actually as a tool that will be there for when you go to get a diagnosis for, let's say, for a rare disease or for inherited disease, that it's easier?
Yeah, so we tested this pipeline with 2 clinical geneticists that didn't necessarily have a background in bioinformatics. And that's also where I realized that we need to talk more. So the people that are behind the computer need to talk more to the people in the wet lab and vice versa, that we understand each other's language and that we also know what we can bring together to actually have that synergy. It can also be used by researchers, but what I really try to do is make my tools foolproof. So I tested a lot on students, but the students are the best way to test these type of approaches. And if they don't have a programming experience, they need to be able to just start such a tool and work with it without having to go through a whole bunch of scripts and setting it up on their own computer. So I try to, what I make it user-friendly, but we didn't test the user-friendliness. But I think it can be used for both scientists and the clinical.
And just for me to understand, because you are using platforms that are open access, which I think is great and beautiful, but how do you do your quality control because everyone can put what they want there and then you want to use it for diagnosis? Is that a challenge?
Yeah, so actually we used Wikipathways and Wikidata, which is built on top of the Wikipedia model. And often when people hear that, they get a bit scared, because in Wikipedia, anyone can change anything. But that's actually something that in Wikipathways and Wikidata, but also in Wikipedia, there are people checking. And they have a lot of automated tests to detect weird behaviors. So if someone is adding random website links, alarm bells goes off. But this is also something that we try to do with this community creation approach. I think the more people look at something, the better the quality in the end will be. And I think that's one of the main reasons to use such tools, because you actually allow people to add the data themselves.
Okay. Thank you very much for your answers. And I'm happy with that, and I give the word back to the co-rector.
Thank you.
Thank you very much for your opposition, Professor Svetlov. Then we come to the following opponent, who's also a member of the assessment committee, and that's Professor Blaak. She's professor in human biology at Nutrium at Maastricht University, and today she has also accepted the role of secretary, for which I'm very grateful. The floor is yours.
Thank you, Mr. Pro-Rector. Dear candidate, and at first I also would like to congratulate you with this very nice thesis and also your promotion team. I read it with great interest. I also learned a lot. And I'm impressed with the contribution of your methods in capturing metabolic pathways and their interactions in blood or at level of specific tissues. And also, your parallel with the SYMPHONIES is quite nice. So the subtitle, Utilizing Graphs as Sheet Music to Orchestrate Biochemical Interactions Relevant in Health and Disease. I think it's very nice, but also very ambitious, I think. And I'm also would like to discuss a little bit with you the application. I'm myself also in human biology, and we also look a lot at inter-organ crosstalk, in metabolism, in health and disease. And although your pathways and also what you added, the metabolic graphs and metabolic fluxes, are very nice and insightful, to what extent can they cover also the complex human inter-organ crosstalk? Are we already there, or can we get there in future?
Highly esteemed opponent, thank you for your kind words and this very interesting point that you bring across. At the moment, the pathway models that I'm using, so the math and the computational part behind the reactions, would not be, I think, the best way to model inter-organ crosstalk. However, I know that there are a lot of changes going on, and a colleague of mine who's also here is working on that we actually can model more than what we are currently modeling. And one of the examples is the diseases that I added. That was something that there was not really a machine-readable manner for. So I made it as text labels, because I had to start somewhere. And at some point we said, okay, we want to do something more with this. We need to make actually a larger model so people can put in more information. For example, microbiome information, if you have different species there. And what we also really need is, where are these reactions happening, in which tissue, and what transport reactions are there. And then in the end, maybe we could link that to the inter-organ crosstalk. Yeah, that would be very interesting, but I know that the technology is not there yet, but it's close.
Oh, that's a very interesting remark. And also, I'm also very interested in the microbiome, and in particular what it does in different tissues in humans, how it interacts. So you think we're close, or
we are close to have the technology ready, but is the data ready? Do we have enough information in publications to actually link this to computational models? Because if the information isn't there, I cannot model it. And I know my paranymph here on the right is doing a lot of work on this with adverse outcome pathways, also going from a small molecular event to a larger organ failure, for example. But also there, the tools are there, but if the data is not there, then it's really difficult to make a computer understand it.
Okay, yes, very interesting. And what, of course, when you look at human research, it's increasingly evident that one size does not fit all. So we have different etiologies towards disease, which may require subgroup or individualized treatments. How do you see the application in the field of precision nutrition, for instance? And then I mentioned a few aspects that may be of importance, for instance, incomplete pathway coverage, lack of personalization, and data integration, of course, very important. So can you give your view on that? It's clear that we're not there yet, although you importantly contributed to the field. But what do we have to do to come at a certain point that we can apply it, or how should we apply it?
Yeah, that's a very good point. I hope that in the next 10 to 20 years we will get a lot closer to this. Especially if we talk about healthy nutrition, if people could monitor their own blood levels, that could help them to change their diet if needed. So I think bringing knowledge actually to the people that would have to implement that diet, that is very important. So we use a lot of open source tools, so it is available for everyone, but it's really written in an academic way. And I think more outreach would be needed actually to the community to make sure that people know what diet, what effect that has on their health. And specifically for the rare metabolic disorders, I think one of the best ways to approach the therapy for them is to change the fluxes. So having that information and comparing that to a healthy situation, maybe we can find a route around a certain reaction that is not working well. But in this, we also need a better coverage of all the metabolites that are there, all the reactions that are happening. And I really hope that in 10 to 20 years, we are a lot closer. But those are the things that I think would be really good contributions. So having the fluxes and doing outreach to the community.
Yes, and of course we would like to be there in so many years. But to what extent is it really necessary that we can model and know all the underlying reactions, for instance, in the field of precision nutrition? Or do we have to go more towards clinical measures or a kind of clustering biomarkers? This can be completely grasp the understanding of all processes in a model.
Yeah, I hope we can at some point, but I also realize that that might just be too much data to work with at the moment. And there was a very recent paper where they tried to model all the reaction paths in the brain of a mouse, and that was terabytes. And that's also where we're running into a lot of issues, that the computational power might be lacking at the moment for this. And I think it also depends on the nutrition strategy. If you're already healthy, then this is a different thing than if you have a rare metabolic disease and one enzyme is not working. Then we need to know, okay, which reactions are before that enzyme and which ones are after it. So I think it depends on the use case.
Okay, thank you very much. I'm happy with the answer, and I would like to discuss it in future a little bit further with you. Thank you.
It would be great. Thank you.
Thank you, Professor Black, for your opposition. Now we come to the following opponent, who's also a member of the assessment committee. That's Professor Szymanski. She's professor in environmental cheminformatics in the University of Luxembourg, and that is in Luxembourg. And thank you for coming all the way from Luxembourg. The University of Maastricht is always glad to have people from outside.
Thank you very much for the introduction, and I would also like to congratulate the candidate and also the supervisors for this amazing work, 7.5 years. And the end is almost there. I would like to go in a little bit more detail on the cheminformatics, so switching from the previous opponents. You've mentioned a lot of identifiers, both in your thesis, but also in your presentation for the audience today. What is your preferred identifier for chemical structures and why? What are the other possibilities and what are the pros and the cons?
Highly esteemed opponents. Thank you for your congratulations and also for bringing up this point. I do not necessarily have a preference, but if I would have to pick one, I would want to use the database that has a lot of chemistry in it. And there are differences between that. So some databases are modeled from the biological perspective and others more from the chemistry perspective. One of the identifiers that's really great for me to use is the INCHI key. because it's short and a computer can read it. But I also know that there are issues that if you have an INCHI key that it's in non-database, you cannot trace it back to the original molecule. But that's more a structural identifier, so that I can link it to structure. If I would have to pick another database, it kind of also depends on what type of molecule I'm looking at. If I'm looking at a molecule that's more in the field of toxicology, I would go to different databases than if it's a human metabolite, for example. But I don't necessarily have a very specific preference.
Okay, just a quick follow-up on that one. You also mentioned names a lot. Can you expand a little bit on why names were so important for you in the thesis?
So for me, I like the structure the best. I want to see the molecule and work with that. But I know that people, especially the collaboration we had with the hospital, they also look at molecules. But then if the information goes to the metabolic pediatrician, they don't necessarily have that full chemistry background. But the names do mean something to them, because that's in textbook that they had to read from start to finish and learn by heart. So I think we need to have the link to both, because just a number for the computer it means a lot, but if I see CABI 1, 2, 3, 4, 5, I don't know which molecule it is. So I think if we can actually go from identifier, name, and structure and show all of that, but to the right person, so to the right audience, we can already win a lot.
Thank you. I would like to ask one of the paranymphs to read out Proposition 9.
The purpose of sharing data should not be to please journal requirements, but to share knowledge with future generations.
Thank you very much. So based on that proposition, I'd like to ask the next question. Could you please summarize for us in a simple manner how databases can profit from using RDF and why they should be provided in this format?
A short answer to that, okay. Because RDFs allow to link data, and then we can find out which information is missing in databases and link it better, so connect it better together. Does that answer your question?
That was a very short answer. Okay. Okay, well then I have time for some more questions. So you also mentioned on page 25 of your thesis that using open science approaches could prevent commercialization. Can you expand on this a little bit? And also, I would be curious why you moved from the CC BY to a CC0 license.
Yeah, so if we already share the data online in a public repository that everyone has access to and can modify at their own will, there is, I think, no point for commercial companies to take that and then store it in a tool that then only people can access if they have money for that. Because the data is already out there, so we are giving it to the community. And it doesn't matter where you're from, as long as you have an internet connection, you can access the data. So I think that is very important. And then your second question was again?
Was why the move from CC BY to CC0?
So that is not necessarily something that I decided. That was decided by other people. But I do think that sharing CC0, so everyone can modify, share, delete, take it into their own tool, gives people even more freedom than the CC BY license would do.
Okay, I know Professor Thiele is going to ask potentially a question building on this, but I just want to dig a little bit more into this. She's going to go into a little bit into incentives. But if we have a CC0 license, people don't even get the attribution. What is their incentive then for contributing to some of these open resources?
Yeah, so that's a good question. So people do not need to attribute, but they can. Mm-hmm. So there is actually no limit to give credit to people that actually worked on the data. I think also in Wiki Pathways, it's very well listed. All the authors that worked on the pathway get credit in that way. And sometimes I look back at pathways that a student that I supervised 4 or 5 years ago made, and I see their name and I recognize those people. So I think this is more a bottom-up thing. We need to actually make sure that people do give credit for the work that people are doing, and not take the data, use it, and don't say where it came from. I think also the provenance there is very important.
Yeah. I'm going to return to the first part of your answer. So preventing commercialization, if the data is open, do you have anything against commercial tools being able to use that data to provide their customers better service if they have the open data plus the closed data as well? Because this is an issue we also fight quite often.
Well, the license gives them that permission, so they can. What would be nice is that they actually maybe contribute a little bit of their closed data, so everyone can benefit from it. And I understand companies need to make money some way, but if the knowledge is already there, it's published, and this is paid by, for example, European grant money, then that's what taxpayers are paying for, right? So we want to see what is coming out of that, and then hiding it behind a paywall is actually kind of sad, I think. Mm-hmm. And if they add something with their own data that they don't want to share or cannot share, that can also be the case, then good for them, and they can use our data as well. But if they are willing to share, then everyone can benefit from it.
Do I have time for a very brief follow-up?
Yeah, short question, short answer.
Just, yeah, exactly. How will you maintain the developments such as Wiki Pathways and especially BridgeDB beyond your thesis?
Well, I still have a contract of about a year. And we are working to automate everything so that the building of these tools and the software is happening without us touching it. And that we also get a message if something is broken. I do think that Wiki Pathways has been around quite for some time, and there are always new people working on it. And sometimes I'm at a conference, I see someone that says, I use Wiki Pathways. I'm like, oh, great. You know, but that's also the message that we need to get out there, that the tool is there. And the more users we have, the more sustainable I think this tool will be.
Great. Thank you very much.
Thank you.
Thank you very much for your opposition, Professor Szymanski. Then we come to the following opponent, who's online, and that is Professor Thiele. She's Professor in Systems Biomedicine at the University of Galway, and that is in Ireland. And thank you, Professor Thiele, for coming all the way from Ireland, albeit online, but more than welcome, and the floor is yours.
Okay, thank you, Mrs. Slenter, for a very interesting presentation and an excellent thesis. And also congratulations to the promotion team. I have a few questions, and one is a principal question on what is enough? Like, we have the mass spec data, of course, we measure metabolites. There are new features that are discovered or which haven't been annotated yet. So what do you think about the comprehensiveness of databases such as Wiki Pathways or others? You did a lot of work during your thesis on expanding. Is there a lot missing? Are there certain pathways? So what is your experience, and what do you think are the ways going forward?
Highly esteemed opponent, thank you for being here, although it's online, but I understand Ireland is even further away than Luxembourg. And also your question is a very important one. I believe that we don't know how many metabolites are out there. So putting a number on how much is missing is very difficult. We can give a rough estimate for humans or similar species. However, we also eat, and we eat salad. Not everyone eats salad, but a lot of people eat salad. And then we eat plants, and plants have even more metabolites in them that enter our system. Where are they going? So putting a number on what is missing is difficult. What I did notice when I started was a lot of the reactions for the metabolic disorders were not covered in Wiki Pathways, as well as lipid metabolism. And that's also what I address in chapter 3 of my thesis. And lipid metabolism is also just very difficult because the annotations of the lipids are often on a class level and not on a very individual species level.
Following up this question, on the one hand, of course, there are the metabolites, but then there's the reactions. So how much biochemistry do you think? And again, I don't think of numbers, but more maybe subject areas. How much biochemistry you think is still discoverable? Let's consider microbes where many of the potentially metabolic enzymes still have unknown functions. We know that they're biochemical, but we don't know what they do. So I suppose it's a tricky question, and yet you have a lot of experience over the last few years in, what is new biochemistry that we might discover? Have you discovered most of it?
I think we are quite far in that aspect. What I think is still missing is specifically for these rare metabolic disease patients, when one enzyme is broken, sometimes the body finds a different route that we didn't know before existed, but our body tries to get rid of a toxic substrate if it's there, in any way, shape, or form it is. And that's actually a way also for us to discover new metabolic paths. What we don't know, what we do know, I think still testing these metabolic reactions in the lab, especially if we go from one species to another, for example, a modeling species to the human situation, can still give us quite some novel insights that we didn't know before. So I do believe that the wet lab is not there yet, and we still require quite a lot of biochemical tests.
Okay, thank you. This kind of expresses the idea of moonlighting enzymes or proteins. And how much does WikiPathways does cover moonlighting enzyme functions?
Excuse me, could you repeat the question?
Do you know to which extent, either through your work or in general the work done on WikiPathway, how much moonlighting functions or secondary functions of enzymes that might be discovered within the set of inherited metabolic diseases or other contexts are currently covered?
For that, I cannot put a number on it. I know that there are proteins with some of these functions, but we didn't classify it as such. So we only link it to the metabolic reaction that is known. What would be very interesting is actually to test this with data to see, does it maybe have another function that we are unaware of? But I am not sure if there is a database out there that has that information. So if you have any insights or tips on that, then I know what to do the next year or so.
Sure, I can think about that. I'm not aware of a database either, but the knowledge is partially there. The question is, does it really occur in, let's say, human, or does it not occur? And that's the tricky part, and there I agree, experimental data is needed. Another question is, during your thesis, you probably had a lot of struggle, headaches, good moments, bad moments, to go between one database identifier and another to kind of, you mentioned the harmonization of reactions and metabolites. I'd be interested in understanding what are the biggest challenges that you encountered and the lessons learned going forward.
Yeah, so that is a very important topic indeed to address. A tool that's developed by our group, BridgeDB, takes care of part of the mappings. However, a lot of issues arise when we have a very similar structure and one database gives it another name than another one, and the matchings are from one database to another there, and from the other one they're not there. Another point here is stereochemistry. So how detailed is the knowledge on the stereochemistry? And the last part is the charges. So if a molecule is drawn with a slightly different charge because of a different pH, then by another database it might not be recognized as a direct link. While in the pathway and reaction models we have, these things are linked. So those are some of the things that I encountered and still haven't figured out completely how to resolve, unfortunately.
Okay, thank you. I may have time for a very brief question, and Professor Szymanski alluded to that, is how can we keep community engaged? I know that there are the names on the WikiPathway where you can say, okay, I contributed to those many different pages, but at the end of the day, citation count, publication count, if you're a PhD student or a postdoc. So what kind of ideas do you have going forward to make community engagement really successful and ensure the longevity of databases like WikiPathway?
Yeah, so I believe that numbers here are important. So what we are actually doing for the new WikiPathways website is counting how often is a pathway cited in literature. We are not necessarily linking that now to the authors, but we could make a rank of which one of the authors' pathways gets cited most, try to go back to the authors that actually use their pathway models and ask them, why did you use this pathway model? Was it because you liked the visualization? Was it because it just popped up in your data analysis? What are the reasons for that? If we understand that better, then that might help us to aim our research and visibility in certain directions. I've also attended a biocuration meeting once, and there they really interacted with the community a lot. And we also try to do that, but we are also depending on how much time people have to put in information. So they need to get credit back for that indeed. And citations is one manner there.
Thank you very much.
Thiele, for your opposition. Now we come to the following opponent, who's also a member of the assessment committee. That's Dr. Cavill, and her expertise is data analysis in the Department of Advanced Computing Sciences in the Maastricht University. Dr. Cavill.
Dear candidate, thank you for the opportunity to read your thesis, and congratulations to you and your supervision team on a very nice piece of work. I'd like to begin by asking one of your panelists to read Proposition 6.
Fact-checking of results obtained from machine-read data requires a human in the loop, now and in the future.
So we have seen over the past several years that AI can do many amazing things that 5 years ago I'd have said were decades off, if not impossible. And we also see that some of the modern AI is prone to hallucination and things that you wouldn't want in a fact-checking task. But at the same time, we see other AIs, for instance, IBM Watson, that was designed to play Jeopardy in the US, that really was able to mine facts found from across the internet and use those facts to answer questions and win the Jeopardy television quiz in the US. So what is it about this task that you believe we can't build an AI that we're always going to need a human in the loop?
Very esteemed opponent, thank you for this question. I wrote down this proposition to be a bit provocative, and that's probably why you asked about it. So even though there is a lot of talk about artificial intelligence and large language models, I often say jokingly, well, AI, it's just machine learning, and machine learning is just fancy statistics. To make a bit of a discussion with people working on this, the reason why I think this is still needed is indeed because of the hallucinations. And being right out of 100 questions for 90 questions, yeah, what about those other 10? If those are patients, you know, in a quiz like Jeopardy, doesn't matter if you have 10 incorrect. But if you have 10 diagnoses incorrect out of 100, it's quite a lot, and you don't want that. Those are human lives. And especially for the rare metabolic diseases, the data is so scattered and so scarce, and these large language models really depend on a lot of data. So there, I don't think it will work that well. And also, I think there is a famous saying that there is no model that's completely correct. And maybe in the future, there will be models that are close to being perfect. But then what about the novel biology that we discover that is not in those models? What are we going to do with those variants of unknown significance that we still need to do wet lab tests to actually verify this?
So one of the things that often comes up in this debate around using AI or having a human is, yes, the AI is going to make mistakes, but humans make mistakes too. At what point can we switch to, or is it just better to always have an AI and a human together because they make different mistakes?
I think that's a very interesting point. I think the 4I principle in general would be better, than not one person looks at the data, but at least two, and that are unaware of this, that they are both doing this, and then compare their findings. And if they agree, then you say, okay, this is probably more likely to be true. But if they disagree, then maybe a third person needs to be involved. So also there, I think the community aspect can be very important, but with the right expertise. And also here, maybe the combination of AI and then a human can be very interesting. But I think the human needs to make the decision. We cannot tell patients, yeah, the algorithm told us that we cannot give you this drug. Why? Yeah, we don't know, it just told us, so we're going to trust it. We would always need to verify and check and try to understand what an algorithm and why it is telling us something.
Yep. Then I want to switch over to chapter 9 briefly. In chapter 9, you were visualizing the patient data in the pathways, and you comment that it's not always possible to visualize patient data in a single pathway. And you said you were being provocative with your previous proposition, so I'm going to be a little bit provocative now. Pathways, I believe, are human constructs. They're arbitrary splits of the full metabolic network. So why would we expect that a single pathway could capture all that information from a single patient? Or perhaps you disagree with my arbitrary splits statement on pathways to start with.
No, it does sometimes feel very arbitrary. Like the figures I showed, those are real figures when I typed in the word GABA. Metabolic pathways, they all look completely different. They start at a different point, they end at a different point. But what I do realize is that people in the clinic, they are trained with pathway knowledge. And for them to understand what we're working with, we need to give them a construct that they are familiar with. If I show them a network visualization with 500 nodes, they don't really know where to start. So that's also what I try to address in chapter 4. We do merge everything together, but then we take out a subnetwork, and in that subnetwork we do connect the pathway names, because that is something that is a common construct that biologists and chemists and people in the clinic understand. They know that, and that can help them with the interpretation. So that's why we try to still use these pathways there, but that doesn't mean that that's the best way to go forward, but it's a way to be practical in this sense, I believe.
And you mentioned that the GABA pathway you pulled up, you had different versions. Is this a big issue in terms of how the clinicians are trained, if they're trained with pathway knowledge? I mean, I remember looking at the apoptosis pathway from 10 different databases, and it was completely different in every single database, and there were only something like 2 genes that were in common across all 10. So what does it mean if they know the apoptosis pathway? Which do they know, and how do you discern that?
Short answer, please.
Sorry.
Yes. That is actually a very important point. So we also, as modelers, need to know what people in the clinic are used to working with. That's also why I used a clinical genetics reference book as a starting point. Short enough?
Good for me. Thank you very much.
Thank you, Dr. Cavil, for your opposition. The following opponent is Dr. Queralt Rosinach. Her expertise is systems, biology, data integration in the Leiden University Medical Center. And thank you for coming all the way from Leiden.
Thank you very much, Prorector, and thank you very much and congratulations for this excellent work and thesis. It's been a pleasure to read. You did tons of excellent work, and also I would like to extend my congratulations to the promotion team as well. And you built actually a very nice symphony, putting all together from curating and capturing new knowledge, making it computational, modeling it, using semantic web technologies to showcase with different applications how to use this in a computable way. And you, as a chemist also, I would like to say you thank you for making metabolites the first-class members in this computational biomedicine world. And indeed, you made a lot of effort in integrating biological knowledge with clinical information, and making and modeling this as in a graph manner and enabling graph analytics. So I would like that you maybe explain us more about the difficulties that you encountered integrating clinical knowledge into this biological knowledge, and suggestions to the ontologist community, how to improve our ontologists for making data integration better.
Very esteemed opponent, thank you again for traveling here and also being present during the mini symposium this morning, where we also addressed things in this sense. So one of the people I collaborated with gave a presentation there, and actually what she realized is that the data that is now there in the hospital is not machine readable. They have names for the chemical molecules in Dutch, and that doesn't link really well to anything else. So the first thing was, okay, what do we need? And I said, well, an identifier, this numeric code, because that's something that computers can understand better than names written with different spellings, especially chemical names, with a dash, without a dash, with a space, without a space. There are so many options there that makes the interoperability very difficult. And then linking this to the clinical data was also quite challenging because we only have one patient at a time for one disease. With these rare metabolic disorders, I would love to have more data to work with to actually find more pathways. But metabolomics in that sense is also a bit limited sometimes. We only know the metabolites that we measured. And if we don't have a lot of patients, you're looking for a needle in a haystack of needles and then hope that it will actually match. And that's also something that we realized. We needed to put in a lot of curation work, and actually part of that was done by a student of mine who's sitting here in the audience and now doing a PhD herself. A lot of manual work, and especially with these databases that don't agree with each other, what are we going to put in the model, and how do we make sure that the identifiers for the interactions, because also those have identifiers, do not mix up the identifiers for the metabolites. That's also something that we realized was quite difficult. Yeah, and then at some point I just do a bit of manual curation. I don't think the field is there yet for these diseases to do everything automated, but at some point, hopefully we will have enough leverage to push this forward and to do this in more of a bulk approach.
And what would be your recommendations for the ontologist community?
Yes, that was the second question, sorry. So I think actually making people in the lab more aware of ontologies, and that it would be great if they would annotate their data with ontologies, because then I don't have to do it, because I'm not an expert on what they measured. So I know a lot about the modeling part, and I know something about metabolites, and I know something about proteins, but the data, I don't know what they measured in a lab. So if they tell me that, and for each term I need to link an ontology term myself, that's a lot of work, and we need to go back and forth quite a lot of times. But if this would get integrated into the workflow in the wet lab already, you do not start a new study before you have all the metadata in a structured format linked to ontologies, and there are tools to help people with that are user-friendly, then you're not going to do the experiment. And that sometimes requires a top-down approach, which I'm not a fan of. What I do try to do is teach to a lot of students what other approaches are there and how they can use ontologies and identifier mappings. So maybe when they do a PhD or go into industry, they have that knowledge and they can then show others how that is done. So more the bottom-up approach.
And thank you. And did you encounter the problem of that having to ask ontologies to add new terms because something was not tracked yet in ontologies?
I actually had to do this for this thesis and also for other projects, specifically for the rare metabolic disorders, that I find in papers for one enzyme, different disorders, but then in another database I don't find them. Or the metabolites that are there is only a top-level hierarchical term, and the subclasses are not there, which are biomarkers. So I'm very happy that some of these databases are very open-minded. They just have a GitHub repository. I can make an issue request, and they fix it for me. Not every database does that, unfortunately. And then the nice thing with Wikiposis is people can just add it themselves, right? So they don't need to go through that step to ask for an issue and then let someone else fix it. But yeah, there were quite some cases where the diseases that are known in literature are not in the databases. There is just no database that's complete, and especially the databases working with ontologies, also there's still quite a lot of information missing.
Thank you, great. And another question would be, I spent some quite time now on building knowledge graph and for AI learning. And I was wondering if you have some recommendations on how to make this knowledge graph building, extracting metabolite-related information better using SparkWell queries, or the problems that you face, and maybe giving us some recommendations or solutions to that?
Yeah, so one of the key things here, I think, is identifier mapping. And what we are working on is to use that INCHI key that I already mentioned when Professor Szymanski asked a question, because that construct has information on the structure itself, on the stereochemistry, and on the charge. And if you cut off those two pieces in the back, you only have information on the structure. So you will get more links. You might also get a few false positives. That is true. But this could be a way to actually do that, and then you skip the databases and their identifiers themselves.
Thank you, Dr. Queralt Rosinach, for your opposition. Dear candidate, Ms. Slenter, the time appointed for the defense of your thesis has passed, and the degree committee will now withdraw to discuss the quality of your thesis and the way you defended it. I request that you and your company, online and on site, await the results of our deliberation, and I will return in this room. And with that, I suspend the meeting.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. I know what's on the line. I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. South side, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
U kunt op plaats nemen.
I reopen this academic ceremony. Mrs. Slenter, candidate, the degree committee here present has assessed the quality of your thesis and the way you defended it. And in view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Evelo is authorized to confer upon you this academic distinction in accordance with your Dutch university custom. I invite Professor Evelo to now take the floor.
Wilt u gaan staan?
Dear candidate, I have to first ask you an important question. Do you promise to work in accordance with the principles of scientific integrity at all times? To be careful and honest, transparent, independent, and responsible?
Yes, I promise.
Okay, then. By the authority vested in us by law, and in conformity with the decision of the committee here present, I hereby confer upon you, Denise Nicole Smaragda Michelle Slenter, the degree of doctor, and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree of certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university.
U kunt op plaats nemen.
Beste Denise, I'll skip all these other names now. Dear Dr. Slenter, I'm really happy to be one of the, well, not really the first to congratulate, because you also did this really nice symposium meeting this morning and you got a lot of congratulations there already, but now it's final. And I enjoyed the discussions a lot that were going on both this morning and here. And I think we will want to continue these soon during the reception, because there's so much still to talk about. But first I grant the floor to Egon, who will give the laudation.
Dear Denise, it's a great pleasure to be able to give the laudatio here to you. Congratulations. I wrote some things down. I don't think they will really do justice to what you showed. I'm very grateful to have worked with you, to have been able to supervise you in this work. It's not that you gave me a lot of choice in that respect. You took very serious ownership of the problem and of the whole question and all the problems. And that started basically during the interview already, where you actually did not answer the questions that I had in the time given, but you decided, okay, I'm going to do all of them. Just wait a minute and you get all of them. That showed for me something that I really admired, and since then it has only been going up. So in that sense, that's more or less it. The book, I put it on your desk. I didn't see it for a very long time, and basically you ended up as a chapter author in that book. I think he did really nice work there in making these diagrams in this physical book on paper into something you can compute on, which you've nicely demonstrated as well. There's so much that we can do in the future there, and that you will do in the future. Regarding your work ethics and why I think you will do awesome in the future is you work hard, you work with care, you have a broad overview, you're critical, and you are, as we saw today as well, not hesitant in explaining where your opinions are coming from. All very positive things, I think. That has shown not just in this defense right now, but with various other things. The number of publications that you have, board position in the Netherlands Metabolomics Center, organized many meetings, very active in teaching at various levels. So, yeah, roughly, where are you going next? I don't know. In our recent talks, I'm not entirely sure if you know yourself already, but I think that it can only be wonderful, basically. So, dear Denise, it's a great pleasure to have worked with you and to really worked with you rather than supervise you. You came with your all ideas, and at best, I think I showed you some tricks, some skills that were new to do. But all the academic thinking, the critical questioning, that was already there. So I want to congratulate you with your first symphony. And I think that many more will follow. And I'm also pretty sure that this is not yet your magnum opus. And with that, I'll conclude this laudatio with one final comment, and that is something for in the future. I think every party should have a dinosaurus, and more about that later. Congratulations, and with that I give the word back to the Pro-Rector.
Thank you, Dr. Willighagen, for this nice laudation. Dr. Slenter, also on behalf of the University of Maastricht, I congratulate you with the degree you have acquired, and of course, congratulations also for you. Supervisors, Dr. Willighagen and Professor Evenal. I hope you, congratulations also for family, friends, colleagues, and your parents, of course. They've helped a great deal. Mm-hmm. And I hope you can celebrate it, and I wish you a great career. I would like to thank the members of the opposition for their work, and of course the PDL and the technical support team for making this hybrid session possible. So I'm about to close this ceremony, and I have a few announcements. Ik zal dat even in het Nederlands doen, dat is wel handig, want die zijn aan de zaal gericht. Dames en heren, ik ga u verzoeken dadelijk de zaal te verlaten, want wij maken hier nog een foto met onze Ierse opponent op de achtergrond. U kan dan alvast naar de refter, waar de receptie is. Dat is linksuit, de pedel zal het u aanwijzen, en dan rechtsaf. Daar kunt u alvast een drankje nemen natuurlijk, want wij gaan daarna nog op de trap een foto maken, ook nog. En als u dan denkt, wanneer kom ik dan eindelijk aan de beurt? Het is vrij simpel, u houdt ons in de gaten, maar wij gaan eerst en dan komt u. En zo gaat dat. And having said that, I close this meeting. En we vragen de fotograaf natuurlijk te blijven.